Apical dominance and evidence that auxins control it. Apical dominance is when the apical meristem suppresses the growth of the lateral meristem. The apical meristem produces auxins which cause growth at the apex, the tip, but suppresses growth elsewhere. Evidence 1. Decapitation. Removal of the apical meristem stimulates growth at the lateral meristems. Another meristem will soon become dominant, producing auxin and inhibiting the growth of others. 2. IAA application. If artificial auxin is applied to where the apical meristem is removed, lateral buds do not grow. 3. NPA acid application. NPA blocks auxin transport proteins from moving auxin cell to cell, so lateral buds are no longer inhibited. 4. Stem girdling. Hot wax at 100 degrees Celsius kills all living tissue it contacts. Auxins are actively transported, and so require living tissue to move. This does not affect xylem, which is made of dead cells. Lateral buds below the girdle are not inhibited. Those above are inhibited. Cytokinins work in synergy with auxins. The auxin in the apical bud causes higher levels of cytokinins to accumulate and so encourages growth. The lateral buds have lower levels of cytokinins and so don't grow. Abscisic acid inhibits bud growth. High levels of auxin in the shoot may cause high levels of ABA in the lateral buds, inhibiting their growth. If we remove the tip, cytokinins will accumulate in the lateral buds, and less ABA will be present in the lateral buds, so they will start to grow. Experimental evidence for the role of gibberellins in control of stem elongation and seed germination. Gibberellins are a group of hormones that help plants grow, stimulating cell division and elongation in the stem. Dwarf plant varieties have been shown to have very low levels of gibberellins. This is often due to a mutation in a gene involved in the synthesis of gibberellins. Under experimental conditions, treating these dwarf varieties with gibberellins results in them growing to the same height as normal varieties. Gibberellins' role in stimulating germination and breaking seed dormancy have been shown in many experiments. Seeds of mutant varieties of the Arabidopsis plant that do not produce gibberellins can be induced to germinate if gibberellins are applied. Seeds of certain lettuce varieties that require light in order to germinate can be made to germinate in the dark if gibberellins are applied.